Hi, today I'll be talking about cerebellum and everything from a little bit of anatomy, physiology. This is mainly to give you a rough idea about what's exactly happening, not essentially covering everything. But if you, in case you were struggling with this topic, I hope this video helps you. Starting off with cerebellum, we have cortex and we have the white matter underneath the cortex, and we have gray matter, that is the nuclei, and the deep cerebellar nuclei. That's what these nuclei are. So starting about the cortex, we have different layers coming from outside to inside. That is MPG, as in you're calling monthly G. So MPG, all right. So molecular, Purkinje, and granular layer. Essentially, the layers contain the cells that they're talking about. I'll talk to you about them in a while. I'm talking about the nucleus, the deep cerebellar nucleus. You have four important deep cerebellar nucleus. Starting from the medial most is the fastigial, and the lateral most that looks like teeth, so you call that dentate. And in the middle you have something called nucleus interpositus, that is, it's placed in between these two, so it's called interpositus. And then you have globose and emboliformis, they constitute this one, nucleus interpositus. So you have fastigial, globose, emboliform, and dentate. These are the four main deep cerebellar nuclei, and we've spoken about the cortex MPG. What cerebellum essentially does is we have so many inputs coming from brainstem, spinal cord, and other areas that relay in the cerebellum, and cerebellum acts as a fine tuner, or the one that corrects all the different sorts of inputs. A little bit about the anatomy. So if this is your brainstem, the inputs and the outputs to the cerebellum and to and fro from the cerebellum come through the peduncles, that is the superior cerebellar peduncle, inferior peduncle, and the middle cerebellar peduncle. Notice how the middle cerebellar peduncle is on the lateral side and doesn't form the wall for the fourth ventricle, lateral wall for the fourth ventricle. Talking about afferent and efferent, to make things easier, middle cerebellar peduncle is purely afferent and comes from pons, so that makes things a little bit easier. So now I'll start off by saying that we starting source, the journey, the relay station, and the journey again, and you finally reach the destination. You already guessed that relay station being the cerebellum and the journey being the cerebellar peduncles. That's how we enter and exit from the cerebellum. The starting and the destination is what I'll talk to you about. The starting, we have afferent fibers. The afferent fibers are essentially only two fibers. We have climbing fibers and we have mossy fibers. The climbing fibers mainly originate from the contralateral inferior olivary nucleus of the medulla oblongata. The climbing fibers originate from the contralateral, C for C, contralateral, and inferior olivary nucleus of the medulla oblongata. Whereas the mossy fibers come from multiple sources, M for M, multiple sources of the brainstem and the spinal cord. You don't have to know, but most importantly being the pontine nuclei and vestibular, and from the spinal cord, vestibular nuclei, reticular formation. So these are the starting fibers. And how do they travel? The journey being the peduncles. Superior cerebellar peduncle is purely efferent. To make things easier, this is how we'll remember. There are a few afferent, but we don't have to know them. The inferior cerebellar peduncle has mostly climbing fibers and some mossy fibers, whereas the middle cerebellar peduncle is the largest input. It's purely afferent and it carries only mossy fibers, M for M again. So climbing fibers purely from ICP, inferior, and mossy fibers purely from MCP. So this was the journey. This was the starting fibers. You had all of them start from inferior olivary nucleus, multiple sources, that is pontine, cerebral, reticular, spinocerebellar, vestibular, and reticular, pontine, and all these start through the cerebellar peduncles, that is ICP and MCP. They come to the cerebellum. Okay, now what happens once these fibers reach the cerebellum? Like we've already spoken, we have a cortex and we have deep cerebellar nuclei. So we'll talk about the climbing fibers. one climbing fiber cell ascends up to one Purkinje cell. So one climbing fiber wraps around the Purkinje cell, whereas the mossy fibers go and stimulate the granule cells. Also stimulate another cell called Golgi cell. Golgi cell is essentially inhibiting the granule cell. Ensures there is check-in excitation of the granule cell. It's feedback inhibition. The same way, granule cells ascend up into the molecular layer as parallel fibers. These parallel fibers stimulate two different cells called stellate and basket, which are again inhibitory to the Purkinje cell. If you look at this, one climbing fiber stimulates one Purkinje cell, one mossy fiber stimulates multiple granule cell, which in turn stimulate multiple stellate and basket and inhibit multiple Purkinje cells. This is the point here. One climbing fiber for one Purkinje, whereas one mossy is multiple fibers inhibiting this. These stellate and basket are essentially in the molecular layer. This is what you have to know happens in the cortex. So from the starting of the brainstem and spinal cord, different fibers through the journey through the peduncles, and they come into the cortex, and in the cortex the protagonist is Purkinje. And why? Because this is the only output from the cortex, and that is inhibit GABAergic neurons, and then that's inhibitory. We have the deep nuclei, like we've spoken. It inhibits the deep nuclei. These deep nuclei have efferents: 2, thalamus, red nucleus, vestibular nuclei, reticular formation, cortex. Now this pathway is being inhibited by the Purkinje cell. These excitatory output from the deep nucleus to various parts of the brain are being inhibited by the Purkinje cells. The efferents, again, the globose, emboliform and dentate, exit the cerebellum through the superior cerebellar peduncle, and the vestigial exits through the inferior cerebellar peduncle. Superior cerebellar peduncle goes to the thalamus, and the tract is, no surprise, cerebellothalamic tract. Red nucleus is cerebellorubral tract that controls the ipsilateral motor, and inferior cerebellar peduncle goes to the vestibular nucleus through cerebellovestibular tract and reticular formation through cerebelloreticular pathway. So what is with this entire system of excitation and inhibition? And this is essentially to maintain motor coordination, balance, planning, feedback inhibition, and fine-tuning of movements. So I hope this kind of makes sense and it's all put together, so you have a rough idea of what's happening and what's not. That's pretty much it. I hope this helps. Thank you.